Welcome, Mindsetters, to this exciting session. I know I'm always excited, but it's grade 10 life science today with the awesome Kathy here. Kathy?
And I'm with the awesome Kai.
Oh, I'm blushing now, if I could blush, but that's a whole other story. Yes, tell us what we're doing today.
Okay, people, we welcome to all the grade 10s. We are going to start with animal responses to the environment, to the endocrine system, which is actually the grade 12 stuff, which means I've opened the wrong thing onto the board. We're the grade 10s. We're going to be doing transpiration.
Okay, so guys, while Kathy makes her way to the board, I'm going to tell you what you need to do, as I always do. Make sure, guys, that you get to the page. You know the drill, www.facebook.com/learnextra. Make sure you get me your questions so I can get your questions to Kathy so I can help you guys out. If I can't help you guys out, then, see, you can't help yourselves out if you don't ask questions. So, you know what? I'm going to now stop and get off my soapbox and actually hand over the lesson to the awesome Kathy here. So Kathy, take it away.
Thank you. Well, Ty,
Yes.
are you going to do a dance for us today, by the way?
I was thinking about it, but I was told last time that, okay, it was a bit too much, so I can't do it again.
It was too short. It was like a blink of an eye. People, you'll notice today, check this out. I am armed
with an
for
explosive
education.
character.
Yeah. Okay, enough nonsense. Let's get down to the structure and uptake of water in plants. Not very exciting, Ty. Geez, you get so excited about this. Yay. Okay, we are going to be going through this. Now, as usual, what I've done is I've put slides in place for you, and they've made little notes of this. I think they call them smart notes, which you can get if you go onto the Mindset Learn Extra website. Okay, but this is so that you know what is going on and so that we can actually check things all the time. Now, why would a plant need water? Think about it. It needs to get this water from the soil into the leaves. And that's the place, you can think of the leaves as the kitchen. And in that kitchen, it makes the food for the plant. So it needs to get the water, which is one of the main ingredients, to the leaves. Why? For a simple process of photosynthesis. Now, just to recap, photosynthesis is the process whereby a plant containing chlorophyll takes energy from the sun, water from the soil, okay, and carbon dioxide from the air, and puts those three ingredients together to make food or nutrients for the plant, and the oxygen is released as waste. Right, so very important process, because that is why we as animals are able to breathe. We need plenty more oxygen than a plant does. Okay, plants do breathe, by the way. They do need oxygen for cellular respiration, but they don't use a lot because they're not very active. We are very active. We need that oxygen, but we also need the plants to use the carbon dioxide that we are exhaling. So we need this water to come up so that the process of photosynthesis can take place. Now, without water, and I mean this is logic, you don't have to be Einstein, no water, no photosynthesis. It would be like if I was cooking breakfast and there were no eggs, I certainly couldn't make an omelette or fried eggs for breakfast. It depends on what there is. You cannot produce nutrients, or a plant can't, if it doesn't have the water. Why? It needs the hydrogen and it needs the oxygens. Okay, so the hydrogens and the oxygens are very, very important. But more than the oxygen is the hydrogen, because carbohydrates, if you recall, are CH, and that would be CN, H2N, and ON. That's your formula for a carbohydrate. So we must have the hydrogens. And what? It takes the carbon dioxide, and from the carbon dioxide, you have the oxygens and you have the carbons. And the oxygens from the water and the excess oxygens from the carbon dioxide are what are released into the air by the process of photosynthesis. All right, so why do we need that water? For the hydrogens. Because if there are no hydrogens, can we make a carbohydrate? Can we make proteins? No. Okay, so very important. Now plants need to get this water up the plant, up the stem to the leaves. So from the roots to the leaves. And why? Because the leaves are the kitchen. Okay, the leaves are the kitchen. And how does it do it? Remember the xylem. The xylem carries water up a plant only. Then, once the nutrients have been produced, they're going to move up and down the phloem to be stored in either the roots or the stem or the leaves. Okay, so when we eat leafy plants like, I don't know, lettuce, spinach, whatever, we're eating the nutrients before they've been transported up and down. When we eat bits of stem like in celery, guess what? We're getting the nutrients which are stored there. And the roots, if we eat a carrot, guess what? That's where the nutrients are. So people in all plants, make sure that you understand that the nutrients are produced in the leaves and then transported up and down the plant in the sap, which passes through the phloem to all the parts of the plant. Okay. Now, if we look at how water moves through a plant, okay, because remember, this is important. We're trying to take it from the roots to the leaves. And between the roots and the leaves, we have a stem. Okay, there are three processes, and these are your three processes: root pressure, capillarity, and transpiration pull. Okay, now we're going to do the whole root pressure thing. Okay. Now, the fact that it's called root and it's called pressure. Now, what is pressure? If I take my hand, okay, and I squeeze this hand, all right, that is pressure. I'm creating pressure. Okay, now, how does pressure happen in a root? You think, hello? I mean, there's nothing to squeeze there. There's, what kind of pressure can there be? Have a look at this. Now, you've done the root hair. So we're going to start with our little root hair. And another epidermal cell. Now, remember the epidermis, just as a recap, these are your epidermal cells. And we have our root hair. And root hairs are, root hair, it's only one, root hair. And a root hair increases the surface area for absorption. Why? Because it extends like it does, it sticks out. So it's a modified epidermal cell. Then, it has, all your epidermal cells here have thin cell walls. Okay? Why? Because you don't want a thick wall, you want a thin one, so the passage is quick, all right? So it has a thin cell wall. And you know this because you've done it. It increases the surface area because of this extension. It has an incredibly large vacuole, okay? Why? Because that incredibly large vacuole contains cell sap. And if it contains cell sap, it is always going to have a lower water potential. Let's go green. A lower water potential, and you will have here the soil particles, you'll have a higher water potential between the soil. Always. Because of this vacuole with lots and lots of cell sap in it. And something else, the last little structure versus function that you must know about the root hair or your roots is that there is no cuticle. Why? Because a cuticle is waxy, and if it's waxy, its job is to stop water from moving backwards and forwards. Okay? Normally you'll find it in the leaves where we want to stop water from being transpired and water loss, excess water loss. Here, saying we don't want it on the root. What's the root's job? Is to absorb water. If you go and put a raincoat on it, which is if you have a waxy cuticle, guess what? The water won't go in from the root. So always a higher water potential than in the epidermal cell. Now have a look at this. We go cortex cells, and I'm just going to do a few here. We have our cortex cells, and what are we going to have with our cortex cells? Guess what? They have large intercellular air spaces. Okay? And what does that do? It helps the water to move along. And as this water now travels through, we then get very special cells, and we've done this, and those special cells here is your endodermis, and that endodermis has a thick strip around it called the Casparian strip. And what does the Casparian strip do? Directs the water. And then we go to the xylem vessels. Looks like a little honeycomb. Okay, so we've got our xylem vessels. Okay, I can't draw in 3D, but that's pretty much what they're going to look like. Okay, thick cell walls filled with lignin, no cross walls, and all the structure versus functions which we're going to do just now. So, what is root pressure? Because all I've done here is drawn this process and show you now. Root pressure, and I hope you're going to be able to see this orange, is the water moves from a higher water potential in the root to a lower water potential. Can you see the red? I'm just checking with my dearest producer. Okay, so the water now moves into the root hair, and from the root hair, because this root hair now has a lower water potential than outside in the soil, it's going to move into the root hair. But once this root hair cell now fills with water, it has a higher water potential than the neighboring cells. So the water will either move through the cells and into the xylem, or it'll move into the root hair and along the cell walls and through the intercellular air spaces to the xylem. Either way, the water is going to move from a higher water potential to a lower water potential. It's like having a queue to go to a concert. And you've got this long queue outside, and as people go in, the whole queue moves up. And that's literally what we've got here. One water molecule will go in, and it'll move along to the next cell, and that means the next one can come in and move along there, and the next one. And because you're going from a higher to a lower water potential each time, and then that becomes higher, and this is lower, and so on, the water moves across. And that pressure that's created by the water moving from a higher to a lower water potential all the way through, okay, that process is called osmosis, if it goes through the cell membranes and through the cells itself, or if it goes via the intercellular air spaces on the cell walls, then it is diffusion. All right. But the water will move from where there is more to where there is less. Okay. And water can never, ever, because of this continual movement from the root hair to the xylem, water will never, ever, ever leave the root hair and go back into the soil. Not ever. Okay, so please remember, it doesn't matter what you put in that soil. It doesn't matter how dry the soil is. Water will, look at this, never, ever move from the plant out of the root hair into the soil. Not ever will that happen. It always moves from the soil towards the xylem. And if there is no water in the soil, in other words, if you've forgotten to water this poor plant, okay, or if there's a drought, then water won't move in, but no water is ever going to move out. All right, very important concept for you to understand. Okay, how are we going for time? Okay, we're still cool, I think. Okay, right, root pressure. And this, all this is in writing what I've just told you. The water potential is higher in the soil than in the root hair, so water moves by end osmosis. End osmosis means to go in, exosmosis means to move out. Okay, so end osmosis, the water moves into the root hair. The root hair has a higher water potential than the cells next to it, so what happens? The water moves from the root hair into the cortex parenchyma, then through the endodermis, then through the Casparian strips. Now remember the Casparian strips? Let's just quickly recap here. Your Casparian strips is, if this is your endodermal cell, okay, all right, you've got your Casparian strips, which are like that, and they prevent the water from going up and down and sideways. It directs the water from that direction through here to the xylem. Okay, so it directs that water. So the water can't go messing around. It says, hey guys, there's a passage here. You're going to go through this passage because your destination is the xylem, not to go podding everywhere else, straight and through. We then move down and we say, right, the cell to cell, and this is very important for you to remember, so I'm going to take it and I'm going to make it bright yellow. From cell to cell, we call symplastic pathway. So that would be a form of osmosis. Why? It's moving from cell to cell. It's, in other words, through the cell. And if it goes through the cell, what have you got? You've got this process of it moving, or the water moving through a membrane. And remember, what is osmosis? Osmosis is the movement of water from a higher water potential to a lower water potential along the concentration gradient, and it must move through a membrane. Okay, you never ever have, just like you don't have water leaving the plant to go into the soil, you never ever ever have a situation where water will not move through a membrane from a higher to a lower water potential, and you can never ever ever reach an equilibrium. You reach an equilibrium with diffusion, never osmosis. Why? Because of that membrane issue. All right, so cell to cell or symplastic pathways is a form of osmosis, or it goes along the cell walls, never goes through the membrane, and we call that apoplastic pathways. Okay, symplastic, it sympathizes with the cell and it goes through the membrane. Apoplastic, around. Okay. Some water moves intracellularly, that would be symplastic, and others move intracellularly via the air spaces, and that would be apoplastic. All right, a high water potential develops in the root xylem because of the force of root pressure. That root pressure is, if I've got a queue and they really want to get through the door, as one person pushes through, a whole bunch of people push forward, and there's that pushing to get in. That's the water molecule pushing to get into the root hair all the time. All right, and then once it's in the root hair, to get to the xylem, there's that cue that's pushing from the back all the time because more people are coming in. So more people have to move forward. And that's what the water molecules do. And if you can picture that in your head, you've got root pressure taped. Okay, you know what? I think before we go on to capillarity, which sounds very, very complicated, actually it isn't, it's so simple. I'm going to give you over to Ty, dearest, and he's going to take us to an ad break.
Yes, thank you, Kathy. I don't know, Kathy always feels like this super awesome mom to me. But anyway, so I said, guys, make sure you stick around. Make sure you just go grab a little snack, go grab something to eat, so you can make sure you have like a little snack, so you don't have to keep going up to the bathroom or to the kitchen, so you get distracted, so you can pay attention. Because as she said, this is a very important section. So we're going to see you after this break. Welcome back, Mindsetters. I hope you've had a nice little break and you're now ready to go and you're ready to pay attention. You've got yourself a little snack. You went to the bathroom. You sorted yourself out. You don't have to get up anymore, so you can pay attention because that's what you need right now. And guys, please, please, please get on the page and chat to me. Let me know what you're thinking. Let me know if you're lost anywhere, if you need help, so I can get those questions to Kathy so she can help you. But for now, I'm going to hand over to Kathy. Take it away.
Thank you, gorgeous. Okay, and Kuni, you are such a star, doll. Thank you so much for helping us. I think next time I'm in Cape Town, or I intend to get to Cape Town, which is probably in the next 2 or 3 weeks, I'm going to email you and maybe we can sit down and have a cup of coffee. But thanks for helping our learners. You're a darling. Mwah, mwah, mwah. And I hope the learners will also send you little kisses because it is awesome. All right, so thanks, hey? Okay, people, just to recap, structural suitability of a root hair. This is, in the old days, used to be a question that was in every test, in every exam, and I promise you now, well, if I was a betting person and I was into gambling, I would give it 100 to 1. This is it, or 100 to 100. What's it? If it's good, Ty, if you're betting, I don't know these things. A 1 to 1. Okay, in fact, probably a 1 to 2 chance that you would get this. All right, so it's probably a 99.9% chance. Now, don't go tell your teachers I said so, because otherwise they won't put it in deliberately. But this is a definite, as far as I'm concerned, a definite question. Structural suitability of a root hair. It goes to structure. Structure versus function. Okay. It looks like, because it does. So, root hair, increased surface area. Why? For efficient absorption. Thin permeable cell walls. Why? Unrestricted movement of the water. I mean, people, this is so logical. Has a large vacuole. Why? So we can maintain that concentration gradient from a high water potential to a lower water potential. And more important, no raincoat. Okay? No raincoat. No waxy cuticle. So for structure versus functions, this can be anything, either 4 marks or even 8 marks, because 1 mark for the structure, 1 mark for what it does. 8 marks in the bag. Okay, now capillarity. I love this word. It sounds so neurologically stimulating. It's Ty. He's a bad influence. He really is a bad influence on me. Okay, capillarity is like synergy. It's like working together. It's like common sense. All right. How many times have you sucked liquid through a straw? Okay. Now the next time, before you suck any liquid through a straw, like a cool drink or whatever, have a look when you stick the straw into the glass, how high the liquid goes above the level in your glass. And I'm sure you'd never bothered to look at that. That is a scientific fact. And the narrower your straw, the higher the liquid will go. Now, look how clever this is. And, you know, we all think plants are doof, you know. I mean, they're plants. I mean, they're like vegetables. They're like plants. How intelligent can they be? Except if you're Prince Charles and you talk to the plants. But I don't know if that makes any difference. Okay, so here we go. The water enters the xylem and it passes through the vessels and the tracheates, blah, blah, blah. The force called capillarity takes this up the xylem. Capillarity consists of cohesion and adhesion. Cohesion is a force where water molecules stick to each other, and adhesion is where it sticks to the side walls, and blah, 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 blah. And you say, okay, blah, blah, fish paste, what does all this mean? I'm going to show you. If I take a trough, and in this trough, I put liquid. And in this liquid, I put a cylinder that's that big, a cylinder that's that size, and a cylinder that's that size. So, actually glass straws, okay? There's my level. Okay? And here, it'll go slightly higher. And here, because, or let's make this a little bit more pronounced. And here, because this is so narrow, we're going to have the liquid go all the way up to there. Why? Because there are two forces. One force is water molecules sticking to now, stick to side of narrow tube. Think about this. If I wanted to climb, let's say I was stuck in a room, okay, and there was this air conditioning chute that's about this size. I could climb up that air conditioning chute to get out of the room. To imagine I'm trapped, I can't get out, I've got to find a way to get out, problem solving 101, all right? I'm going to get into that tube, I'm going to wedge my back against the one side, and, because remember I'm going up, not sideways, and I'm going to use my feet and I'm going to push up with my toes and then wedge with my butt and my back. And then push up with my toes and wedge with my butt and my back. Why? Because I can actually climb up this pipe quite easily because I can fit, and I'm sticking to the side of the wall, okay? That is exactly the same as this force where things stick to the side of a narrow tube, okay? It's called, adhesion. And if you can't get that in your head, think of glue. What does glue do? I can take one side and the other side, and I put glue on, and the glue becomes an adhesive. Sticks the two walls or the two sides together. That's another way to remember it. But you have to know these terms. So, the fact that the water molecules stick to the sides of a narrow tube, okay, is called the force of adhesion. Our next force that we look at, so let's just do this here. There's adhesion. The fact that we can see this meniscus, and with water, it climbs up the sides of the tube, okay? And when you do science and you have to do your experiments, when you do liquids, that's the way they look. If you look at mercury in a tube will do this, which tells you that mercury has a very low adhesion force. Why? Because mercury molecules aren't so keen on sticking to the sides. They're more keen on sticking to each other. Water, however, likes to stick to others, to the sides, more than it likes to stick to themselves, or cohesion, so to each other. Now, stick to the sides of a narrow tube, that is adhesion, or they stick to each other. And if they stick to each other, that is cohesion. Now, you must have heard people say, come on people, united we stand, together forever. one for all and all for one. Let's form a cohesive force, a cohesion. Let's have a cohesive group. Cohesion, sticking together. Okay, that's what we should all be doing as South Africans, sticking together. So we stick together so we can be united and move forward. All right, so cohesion, the water molecules stick to each other. Adhesion, the water molecules stick to the sides of a tube. And the fact that when we draw water in a test tube, this would be mercury, and this would be water. If you look here, water sticks more to the sides than it does to each other. Why? Because, and it's because of that reason that you're going to find your meniscus looking like that. Okay, but it is both forces. Otherwise, it would only stick to the sides and not to each other. With mercury, your cohesion is greater than the adhesion force, and that's why mercury will climb up the side, not climb up the sides, but push from the center up. All right, very important. So both forces together ensure the water moves up a portion of the stem. This process of capillarity, which is made up of cohesion and adhesion. Cohesion, water molecules stick to each other. Adhesion, they stick to the walls. You take these two, makes a capillarity. This will get the water. If this is the soil level, it takes the water up the root, just slightly above the level of the soil. Now, when you've got a tree that is 1 meter, 2 meters, 10 meters tall, that getting it just up to that point is already a nice assistance. So we've got root pressure pushing those water molecules in, and therefore pushing, as they come in, it pushes them up. Okay, root pressure. Capillarity pulls it a bit higher and up past the soil level. And then what do we have? Our third force, and this force is the most important force of them all. Where is it? And why have I not got it here? Just hang on, let's just get a blank slide then. The third force and the most important force is this one. Okay, wait, before we get to that clearly, let's do the xylem vessels first. Okay, when we look, I drew the xylem vessels just now. What is interesting about the xylem vessels? Number one, they have an incredibly thick cell wall. And that cell wall is thickened with lignin. It has a large lumen. They are long. Okay, they are cylindrical. Long, cylindrical, narrow tubes. Okay, let's just make more space here. All right. There are no cross walls. Now, what does no cross walls mean? Those no cross walls is what makes them long, cylindrical, narrow tubes, because if these were all the different cells, the cell walls, let's just do this, your cell walls have got thick, we're drawing that from the side. So the cell walls are all nice and thick, and these cross walls that divided each of the cells and made them different cells, they've all died. They're gone. So these are long, cylindrical, narrow tubes. There are no cross walls, and they are also non-living. Why? Because guess what? If there are no cross walls, all the cytoplasm, all the cytoplasm, everything would just be washed away. Why? Because this thick lignin cell wall, okay, doesn't allow because it's waterproof, doesn't allow anything through. And then the last structure versus function is that these long, narrow, non-living tubes with a large lumen and cell walls that are thickened with lignin, all right? These things, the xylem vessels, also have holes. Why? Because the lignin is waterproof, and these holes allow the water to pass from one xylem vessel to another one and then be sucked up, and then water will go from here and up in this one, and so on, because they're non-living and because they are actual tubes with so much lignin that nothing can get through. So what do? They have these little holes, and these little holes are called bordered pits. And they allow for the sideways movement of the water. So when one xylem vessel gets full, it can pass the water to the next one to help it push up. Okay, so that's important with regards to capillarity. And then structural suitability of the xylem, I've just gone through it. It will be in the X sheet, what do they call them? Smart notes.
Exactly.
There we go. The X. Oh, for heaven's sake! Smart notes. The Learn Extra smart notes is what I wanted to say. Okay, but they are non-living, elongated tubes. The tracheates overlap each other, so there's strength. Okay, and you've got the pit cells, which make the bordered pits, which link the cells. You've got the plants. The cell walls are thickened. And that's why the tension of cohesion and adhesion to get that liquid up the xylem. The cell walls are perforated with bordered pits. We have a large lumen, and the vessels are cylindrical. Anything that is rounded is always much stronger than something that has got corners in it. Okay, so think of all the structures in the world that are really strong. They are rounded because there's no point where pressure can be applied. Okay, now to demonstrate the movement of water through the xylem, this is just a little experiment which is actually quite cute. You take a plant which has roots. When you take it out of the soil, you take it with its soil, all right, around the roots so you don't damage the little root hairs. And then you take your little plant and you go and you rinse it ever so gently under a tap and in a basin of water. Now you've got your roots free of soil, because otherwise it makes the water cloudy and everything looks yucky. You want to have it a nice sleek experiment. And you take your plant. These would clearly be the leaves. Let's just get a color we can write with here. They're your leaves. And we have eosin solution. This is like a pink, red, depends on how much you put in. Put a good dose of eosin in. Mercurochrome also works. And food coloring also works. But don't use blues. That's why the red or the pink eosin is nice, because it's a strong color. And you stick your little plant in here, in the water. And you put it in an area where it can get some sunlight, so it thinks everything's kosher. And it will photosynthesize, and the water will leave the leaves by transpiration and suck the water up. And what you do then, you take a very, very sharp scalpel, and you take your leaf, shame the poor plant, you take this plant out, and you zap it through its xylem, okay, right through the stem. You cut it across the stem, and you cut a very, very thin slice, put it onto a slide, and you have a look at it under the microscope, and you will be able to see wherever the xylem vessels were, it'll have that red dye, or this pink, depends on how much you put in, but it will be dyed, and you'll be able to see it. And if you use something like a celery leaf, so if you can find a celery plant, and you use a celery plant for this experiment, you'll be able to distinctly see exactly where the xylem vessels are, because they've got quite nice ones that you'll be able to see with your eye without having to put it onto a slide if you don't have microscopes at school. but you can also do this at home. So it's something nice to do and to be able to see exactly where the xylem vessels are. Okay, now here we go eventually. I knew I had some method to my madness. We have transpiration pull, and this is the most important process out of the three. Remember, root pressure gets the water from the soil to the root hair to the xylem. It's that much, okay, if even that much distance. And then it's got to get, so root pressure's there, but it happens. Root pressure will push some of the water up. Then what do we have? We have capillarity, which takes it a bit higher. So if our plant was 1 meter high, we've now gone root hair, root pressure, capillarity. Now we've got to get it from here to the tippy parts of our plant, which is 1 meter high. That's a hell of a long way, especially if you're a little plant. I would like to see you suck water or cool drink or juice through a straw that was 10 meters long or 5 meters long. Okay, even 1 meter long, you sucking, it's going to start hurting your mouth after a while. A plant does this all the time. So, what are we going to have? We have this process of transpiration pull. And transpiration pull is the suction force that is created by transpiration pull. So, what is it? Transpiration pull is the loss of the water through a stoma, through the stoma on the leaves during daylight. Now, it should be transpiration. Transpiration is the loss of water out of a leaf. And transpiration is not sweating. Okay, not. We sweat. All right? Dogs pant, we sweat. And we have water dripping off our bodies. That's sweating. A plant never sweats. It doesn't have to sweat. You know why? Because it's not warm-blooded. Have you ever seen a lizard sweat? Have you ever seen a fish sweat? No. Because guess what? They are cold-blooded. They don't need to control their body temperature. Only us and birds. Okay? Your vertebrates, your mammals and your birds, we are warm-blooded. We control our body temperature by sweating or not sweating. Okay, sweating, goosebumps, the two. But a plant isn't warm-blooded or cold-blooded. It is no-blooded. Guess what? It doesn't have blood. All right? There is no blood in a plant. There is water and mineral salts, and nutrients, water and mineral salts. And that's it. Okay? It cannot sweat. It does not need to control its body temperature. Please, I hope you've got that in your heads, because this is something that used to bug the living death out of me. You sit there marking matric papers, and you've got some kid who tells you, oh, when it's very dry, water will leave the plant through the roots and go into the soil. And I want to vomit on the spot. Wrong. I actually want to hunt the teacher down that taught that. Okay. Number 2. So water will always move from the soil into the plant. Always. Never out. Okay. That's because of root pressure and osmosis. Water potentials. And then if we look here at the whole transpiration process, plants do not sweat. Okay. Water leaves because the water is being sucked out of the plant, because it's going, water's in the leaf, and the water goes, zhoops. Why? Because there's less water here and more water there, so water moves out. And it evaporates off the plant. As it evaporates, there's a gap there, and there's more water in the leaf and less outside, so it moves out. It's like drying your washing on a washing line. Same principle. There's no sweating. Your clothes don't sweat when they're wet. Ty?
And on that note, that would be really awkward if my t-shirt was sweating on me right now. That would be weird.
Can you imagine?
But anyway, guys, I hope that was a nice session. But make sure we're coming back after this ad break, so stick around. See you soon. Welcome back, Mindsetters. I hope you had a nice little break and you're back and you're ready to pay attention, and you've had your notepads and your pens out and you're making notes, as I always tell you to do. But for now, we've got a lot to get through in a very short space of time, so I'm going to hand it straight back to Kathy. Kathy, take it away.
Yeah, I'm panicking here. I'm panicking here. Okay, people. So, transpiration pull, number 1. Transpiration, water leaving a leaf, okay, or leaving a plant purely because of water potential and diffusion. High concentration, low concentration out. Okay? The force is responsible for sucking water up a plant. Why? Because as the water comes out, it evaporates, there's space and a new one. So think of this continuous queue coming from the soil into the root, up the xylem because of capillarity, and out the leaf because of transpiration pull. All right, or the suction force of transpiration. And please, it is the most powerful of all 3 forces. Okay, now, no, I want next page. So let's go next page, next page. There we go, transpiration pull. And this basically explains it. How does it work? We have the terminal ends of the xylems in the veins of the leaf, through the spongy parenchyma, into the intercellular air spaces, into the substomatal air space. Substomatal means that it's just above the stoma, okay, and out through the stoma, which is the pore, zhoops, out in the form of, look at this, water vapor. Mind you, you can't see that, hey? It's in the form of water vapor. It is in gas form. Okay, this will be posted for you as well. Now, path of water from the xylem to the leaf, and here we go. Make sure you know this diagram. It's actually quite a lekker diagram that your teachers will give you different versions, and they'll ask you questions about it. Remember, symplastic, apoplastic, symplastic through the cells, apoplastic around it. Symplastic is osmosis, apoplastic is diffusion. Diffusion because the water does not move through a membrane. All right, so movement of the water between the cells. Okay, so it goes boom, 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 boom. It comes out of the, what color have I got here? It comes out of the xylem. They're your xylem vessels. Okay, in the leaf, it's your vein. Out of the xylem vessels now. Where does it go? It goes from cell to cell to cell to cell to cell. And if it's going through the cell, what do we have? Symplastic movement. It is osmosis because it's going through cell membranes. And now it goes into the intercellular air space, blah, 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 blah. Here is your substomatal air space. It is the air space just above the guard cells, which we've done, people, and the stomata. Now, the stomata were the 2 guard cells that look like kidneys. And what do? When they're full of water? Why? Because they contain chloroplasts and can photosynthesize. The inner walls are thicker, the outer walls are thinner, and it pulls the stoma open so that lots of liquid can pass out. So once the water goes from here, so it goes from this cell into this substomatal air space. Let's do this here. Substomatal air space. Okay? Once it gets into that substomatal air space, it moves from the cell through the cell membrane to the air space. Once it is here in the air space, and it moves out through the stoma, which is the pore, to the atmosphere, is there a membrane there? No. So from the substomatal air space out through that pore is diffusion. Okay? Diffusion, not osmosis, because there is no membrane from the substomatal air space out through the stoma. All right, that's a nice trick question, especially if you are at an IEB school. All right, this is blah, 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 and it's everything we've gone through. What I do want to say to you is this, just quickly, and I've got like 1 single minute here, and Ty, you're going to have to just bear with me here. If we look at a cell, and the cell wall is there, and the cell membrane sits all nice and flush against this, and you've got your large vacuole. When the cell is turgid, Okay, it means that the pressure from the inside and the contents pushing outwards is equal to the pressure of the cell wall pushing inwards on that content, on the contents of the cell. When that is equal, so pressure out, pressure in, when that is equal, then the cell is turgid. We need water in the plant to give it structure. Okay, over to you, darling.
Thank you. And that was another awesome show. I hope you guys had fun. Hope you guys were paying attention. I hope you guys, well, from what I'm seeing here, you guys had a lot of fun on the page. Make sure, guys, always get on the page, chat. But for me, I'm going to say peace out and see you next time.